Welcome to the deep dive. Today, we're diving straight into a really dynamic area. How digital innovation is supercharging the traditionally lengthy process of pharmaceutical research and development.
Yeah, it really is speeding things up.
We've gathered some fascinating material, and our goal today is to explore a compelling case study that vividly illustrates this acceleration, from the initial spark of identifying a potential drug all the way through to making it efficiently.
Right. And we'll hit on the benefits, but also, the challenges researchers run into. It's all based on actual scientific finding.
Exactly. And for you, the learner, our aim is to give you a clear, insightful understanding of this fast-moving field, hopefully sparking some aha moments without getting too overwhelming.
Precisely. We're moving beyond just theory here. We're looking at real examples of how these digital tools are actually being used to speed up and refine every stage of the drug discovery pipeline. It's a genuinely transformative shift.
Okay, let's jump right into those initial stages then. Early discovery. A cornerstone here is high-throughput screening, HTS. For you, the learner, can you maybe break down what that technique involves?
Sure. So high-throughput screening, HTS, is basically a way to rapidly test huge numbers of chemical compounds. Think libraries with hundreds of thousands, even millions of molecules.
Wow, that's a lot.
It is. And you test them to see if any interact with a specific biological target, something relevant to a disease. It's like casting this incredibly wide net to see what sticks. The goal is finding those initial hit molecules that show some promising activity.
Okay. And we often hear the term druggability. What does that tell us about a target?
Druggability. Yeah, that's a key concept. It's essentially an assessment of how likely it is we'll find a drug-like molecule that can effectively bind to and modulate a specific biological target.
So some targets are just easier than others?
Exactly. Some are just inherently easier to design drugs for because of their structure or how they work. It's kind of like saying some puzzles are just simpler to solve. It depends on what a molecule needs to do to bind effectively, and also, our past successes with similar targets.
That makes sense. So once these initial hits pop up from HTS, how do researchers start narrowing things down to boost the chances of finding a real drug? I've seen references to drug likeness rules.
Right, that's where those rules come in. A really well-known one is the Rule of Five. It's basically a set of practical guidelines that came from looking at lots of existing successful drugs.
Okay.
These rules look at simple molecular properties, things like molecular weight, how fat-soluble the molecule is, the number of hydrogen bond donors and acceptors, stuff like that.
And what do those properties tell you?
Well, by checking those, we can predict if a compound is likely to have good oral bioavailability, meaning can you take it as a pill and will it get absorbed properly into your bloodstream?
Got it.
So these rules act like an initial filter. They help us prioritize compounds from those huge screening libraries, focusing on the ones with a better shot at becoming effective oral drugs. The scale and speed enabled by digital tech, including analyzing these properties, have really changed the game here.
So it's like a digital sieve for the most promising leads.
Precisely. And while these rules are super useful for compounds we already have, it gets even more sophisticated when we're designing totally new compounds virtually before any lab work.
You mean just on a computer?
Yeah.
Oh.
Computational tools let us predict these same properties for molecules that only exist in the model. We can bake drug likeness right into the design process itself.
So you avoid wasting time on synthesizing duds.
Exactly. Avoids investing time and resources synthesizing stuff that's likely to have major absorption or distribution problems down the line.
But it's not perfect, right? The sources suggest even compounds that break these rules can still be useful.
Oh, absolutely. Experienced medicinal chemists, pharmacologists, they can often analyze data even from non-drug-like compounds and get crucial structure-activity relationship information.
Yeah.
SAR, structure-activity relationship. Basically, they figure out which parts of the molecule are doing the work, hitting the target, even if the whole molecule doesn't look like a typical drug.
Okay.
And that knowledge can then guide optimization, leading to new, more drug-like molecules based on those initial findings. So the rules are a great starting point, but the scientists' expertise in reading the data still totally essential.
Okay, moving on from finding the molecule, understanding how it behaves in the body is critical. We're talking ADMET properties now.
That's correct, ADMAT. It stands for absorption, distribution, metabolism, excretion, and toxicity.
Hmm.
These 5 are absolutely critical.
Why so critical?
Well, a molecule might look amazing in a lab dish, but if it's poorly absorbed or gets broken down too fast, or it doesn't reach the right tissues, or crucially causes nasty side effects,
Hmm.
it's probably never going to become a medicine.
Right. And this seems like another area where digital tools are making a big difference, predicting these things earlier.
Absolutely. We now have a whole arsenal of computational tools and what we call computational descriptors.
Descriptors.
Yeah, think of them as ways to turn a molecule structure and properties, its size, shape, charge distribution, into numbers a computer can analyze.
Okay.
By analyzing these numbers, we can build predictive models. How might it be absorbed? Where might it go? How might enzymes break it down? How is it eliminated? And importantly, might it be toxic?
So you can flag problems much sooner.
Exactly. Lets us focus efforts on compounds more likely to succeed and stop wasting resources on ones likely to fail because of a bad ADMET profile.
The sources also mention using in vitro systems like hepatocytes, liver cells to study metabolism. How do those lab tests fit in with the computer predictions?
Good question. Those in vitro systems, like using liver cells, give us a more, well, biological look at how the body's enzymes might break down a drug candidate.
Because they have the actual machinery.
Right. They contain the real biological machinery for drug metabolism. So we can identify potential breakdown products, metabolites, see how fast the drug is metabolized. This is crucial for predicting things like drug interactions.
Where one drug messes with another's metabolism.
Exactly. And for figuring out dosing for clinical trials. The in vitro data then helps us refine and validate the computational models, making the predictions better.
I also saw something about molecular interaction fields, MIFs. What are they used for?
MIFs. They offer a more detailed way to map how a molecule might interact with its environment, like its target or metabolic enzymes. Think of it like feeling the molecular landscape around a drug using different virtual probes.
Probes.
Yeah, probes representing different chemical features, a positive charge, a hydrogen bond donor, things like that. By mapping the energy between the molecule and these probes in 3D space, you get a map of its interaction potential.
And that helps compare molecules.
It helps evaluate how similar two molecules are in terms of potential interactions. Useful for finding molecules with similar activity or maybe cross-reactivity with other targets or enzymes.
Sounds powerful,
Yeah.
but the sources hinted at challenges, especially with aligning the molecules. Why is that tricky?
That's a really important point. To compare MIFs effectively, you need the molecules aligned in a biologically relevant way. Ideally, how they actually bind to the target.
Which you might not know.
Often you don't have that detailed structural info, so you try aligning them based on other things like common structures or pharmacophores, the key bits thought to cause the activity.
But that's less certain.
It introduces uncertainty, yeah. The
Yeah.
less info you have on the binding mode, the harder and potentially less reliable the alignment is. That alignment challenge can be a real bottleneck for using MIFs effectively.
Okay, let's pivot now to a specific example of this acceleration in action. The INFORM-1 study for hepatitis C virus. This seems like a really telling case.
It is, absolutely. INFORM-1 is a great example of how a smart strategy, using what we knew about the virus and efficient trial design, really sped up Hep C treatment development.
What was the basic idea behind it?
The rationale was to combine two different direct-acting antivirals, DAAs, hitting different essential viral enzymes. It was kind of inspired by HIV treatment, where combination therapies hitting multiple targets worked really well against the virus and resistance.
And the two drugs in INFORM-1 were RG71288 and RG7227.
Exactly. They were designed to inhibit different key proteins the Hep C virus needs to replicate. The thinking was, block two different things at once, you get a much stronger antiviral punch and make it way harder for the virus to develop resistance.
Makes sense. What about the actual trial design? Who took part?
INFORM-1 was a phase I trial. It was randomized, double-blind, and used ascending doses.
Okay, can you unpack that a bit?
Sure. Randomized means patients were randomly assigned to get the drug combo or a placebo. Double-blind means neither the patients nor the researchers knew who got what until the end. Ascending dose means they started with low doses in some groups and gradually increased the dose in later groups to check safety carefully.
And the patients?
They were adults with chronic hepatitis C, genotype 1, common type. Importantly, it included people who'd never had treatment and people who'd tried older interferon therapies, even some who hadn't responded to interferon at all.
And what were the main things the researchers were looking at?
Safety was top priority, of course. any side effects related to the treatment. They also closely watched viral kinetics, basically, how fast and how much the HCV RNA levels, the virus's genetic material, dropped in the blood.
It's a direct measure of effectiveness.
Right. Plus, they looked for any signs of the virus developing resistance. And they did pharmacokinetic studies, PK, to see how the drugs were absorbed, distributed, metabolized, excreted, and crucially, if the two drugs interfered with each other when given together. You can actually see the impressive viral load drops in the data tables from the source material.
So what were the key results? Did it work?
The results were really encouraging. All the dose groups finished the study without any major treatment-related safety issues. No dose adjustments needed. Nobody had to stop because of side effects.
That's good.
And the PK analysis showed no significant interactions between the two drugs, which is always a worry with combos. And, maybe most importantly, they saw substantial, rapid drops in HCV RNA levels across all the groups getting the active drugs, a strong antiviral effect.
Sounds like a really positive outcome then. And the source material stresses this study had a big impact on Hep C treatment going forward.
Oh, absolutely. Inform 1 gave strong early clinical proof that an all-oral interferon-free combo therapy with DAAs was not just possible, but highly effective. It was a major turning point.
Really paving the way for what came next.
Exactly. It paved the way for the rapid development of the super effective, much better tolerated DAA therapies we have now. These have revolutionized HCV treatment, changing millions of lives. The speed Inform 1 showed results, using advanced data analysis and efficient design. It was night and day compared to earlier HCV research timelines. Really showed the power of a targeted, digitally informed approach.
Okay. Shifting from hitting the virus directly, let's talk about getting the drug into the patient effectively. Solubility and bioavailability, these seem like common roadblocks.
They certainly are. Huge hurdles sometimes. For a drug to work, it needs to dissolve properly in body fluids to get absorbed into the bloodstream and reach its target. But loads of promising drug candidates just don't dissolve well in water.
Which limits how much actually gets into the system.
Right. It limits their bioavailability, the fraction of the dose that actually reaches the circulation unchanged and could do its job.
So what can researchers do? Are there formulation tricks?
Yes. There's a whole range of formulation techniques to boost solubility. Things like adjusting the pH, using other liquids called co-solvents to help dissolve the drug, forming complexes with other molecules, shrinking the particle size that's called micronization, or making solid dispersions where the drug is spread out in a soluble carrier.
The idea of prodrugs also came up regarding bioavailability. How did they fit in?
Prodrugs. They're basically inactive versions of a drug, chemically tweaked to overcome problems like poor solubility or low bioavailability.
How's that work?
Often you attach a chemical group that makes it more water-soluble or helps it cross biological membranes better. Once it's administered and absorbed, the body uses enzymes or chemistry to snip off that extra group, releasing the active drug where it needs to be.
The source mentioned an example with phenytoin derivatives in dogs having better bioavailability. Can you explain that?
Yeah, that's a good illustration. Phenytoin is an anti-seizure drug, but it's not very soluble. Researchers attach these N-acetylcyclyl groups to it.
Creating the prodrugs.
Right. These modified versions, the prodrugs, showed better bioavailability in dogs, especially with food, even if their water solubility wasn't drastically higher. The thinking is the added group probably helped absorption across the gut lining, maybe by making it more fat-loving, or interacting better with transporters.
And then it converts back to phenytoin in the body.
Exactly. Once absorbed, it converts back to active phenytoin. Shows how smart chemical modification can fix pharmacokinetic limitations.
We've mostly talked about pills, oral delivery. What about other routes? Nanoparticles were mentioned.
Nanoparticles are a really exciting area for drug delivery, especially for more targeted or controlled release. They're incredibly tiny particles made of polymers or lipids, for example.
And the drug goes inside or on them.
Yeah, you can encapsulate the drug inside or attach it to the surface. By carefully engineering the nanoparticles' size, shape, surface properties, you can achieve specific goals.
Like what?
Like protecting the drug from breaking down, keeping it in the bloodstream longer, or even targeting it specifically to diseased tissues or cells. For instance, they're being looked at for delivering drugs right to the lungs for respiratory diseases, or trying to get drugs across the blood-brain barrier for conditions like Alzheimer's.
So digital tools are key in discovery, formulation, delivery. What about the actual making of the drug? The chemical synthesis and manufacturing. Are digital innovations important there too?
Absolutely. Digital tools are increasingly used to optimize chemical synthesis and manufacturing. The goal is always robust, efficient, cost-effective, scalable production of high-quality drugs while minimizing waste.
The sources mention controlling things like nucleation and crystal growth. Why are those tiny details so critical?
Nucleation, the first formation of crystals, and then how they grow, these are super critical for solid drugs. They dictate the physical properties, particle size, shape, even the crystal form, or polymorph.
And that affects how the drug works.
Profoundly. These crystal-level details impact solubility, stability on the shelf, how well the powder flows during manufacturing, and ultimately bioavailability and how well it works in the patient. Digital tools, like modeling and simulation, help scientists understand and control these crystallization processes precisely.
Ensuring consistency.
Right. Consistent production of high-quality material with the right properties. You see lots of examples in journals like Organic Process Research and Development, OPR&D, where computational tools optimize crystallization.
Quality control is obviously huge. The discussion mentioned chromatography and the need for specificity, especially for mutagenic impurities.
Yes, ensuring purity and safety is non-negotiable. Analytical techniques like HPLC, GC, they're essential for separating and quantifying the drug and any impurities.
And specificity means?
Specificity means the method must be able to tell the drug apart from anything else, related stuff. Break down products, process impurities. Mutagenic impurities are a big worry because they can damage DNA, potentially cause cancer, even at tiny levels.
So you need really sensitive tests.
Extremely sensitive and specific methods, yeah. Often down to parts per million or even billion. Sometimes complex sample prep is needed just to detect these trace amounts accurately.
What about residual metal catalysts? I saw metal scavengers mentioned. Why are they needed?
Good point. Metal catalysts, off the palladium, platinum, things like that, are used a lot in synthesis to make reactions happen efficiently. But tiny traces can remain in the final drug.
And that's bad.
Potentially toxic, yeah, even in small amounts, so you have to remove them. Metal scavengers are materials designed specifically to grab onto these residual metals. They form complexes you can easily filter out or remove.
So choosing the right scavenger is important.
Critical. And optimizing how you use it. Process chemists often use systematic experiments, sometimes guided by digital tools for experimental design and analysis, to find the best, most cost-effective scavenger for their process. Again, OPR&D has tons of examples showing how digital tools help optimize reactions and purification, including metal removal. Okay, so we've gone from discovery, formulation, manufacturing. The final critical stage is clinical trials. How are digital tools changing that phase?
They're revolutionizing clinical trials too, really impacting everything from the initial design through to analyzing the huge amounts of data generated.
For example,
Well, sophisticated statistical software is key for designing trials with the right number of participants for reliable results. And defining clear primary endpoints, the main outcome measure, is fundamental. Digital platforms help define, track, and analyze these precisely.
the discussion also mentioned different trial types, like non-inferiority trials. How do digital tools help there?
Right. Non-inferiority trials aim to show a new treatment isn't significantly worse than an existing standard one. They rely heavily on stats to see if the new drug's effect falls within a preset margin.
So software is crucial.
Essential for calculating sample sizes, performing complex analyses. Plus, computational modeling can explore different scenarios and assess the trial's statistical power beforehand. And biomarkers, they seem increasingly common. How are digital tools helping with those?
Biomarkers, measurable indicators like proteins or genes, are getting integrated everywhere. Digital tools are absolutely vital for managing and analyzing the large, complex data sets involved.
How so?
Using sophisticated stats and machine learning to identify potential biomarkers, understand their link to disease or drug response, and ultimately use them to improve patient selection for trials, getting the right drug to the right patient, and monitoring effectiveness.
Finally, the rise of AI and expert systems in healthcare came up. What's their potential impact?
Huge potential. AI can help optimize trial design, predict patient dropout, personalize treatments within trials. It can analyze unstructured data like health records or images to find eligible patients or insights you'd miss otherwise.
Beyond trials, too.
Yeah, AI is being explored for diagnosis, treatment recommendations, predicting outcomes. But it's important to remember, the regulations for AI medical devices are still evolving. Careful validation and ethics are key.
It all sounds incredibly promising. But the outline also mentioned challenges with adopting all this digital innovation. What are the main roadblocks?
There definitely are challenges. Data integration is a big one.
Hmm.
You've got massive, diverse data from discovery, preclinical, manufacturing, clinical trials. Getting it all together in a standard, usable way is complex. Needs robust systems.
Quaz.
Well, you need robust, reliable computational models. Their accuracy depends on good input data and sophisticated algorithms that requires constant development and validation. And then there's the upfront investment in infrastructure, hardware, software, data storage. Plus, crucially, training people to use these tools effectively.
But despite those hurdles, the overall direction seems clear. More digital acceleration.
Absolutely. Despite the challenges, the trend is overwhelmingly towards adopting more digital tools across the board. The potential benefits, faster timelines, more efficiency, lower costs, and ultimately, better, safer drugs for patients are just too significant to ignore.
Well, this has been a really insightful deep dive into how digital tools are fundamentally reshaping pharma R&D. To quickly summarize for you, the learner, we've seen how these innovations speed up initial drug discovery, optimize preclinical studies, streamline manufacturing, and transform clinical trials. That HCV case study, Inform 1, really highlighted the potential for rapid progress when you combine scientific insight with digital power.
Yeah, it's clear digital innovation isn't just some future idea in pharma R&D. It's happening right now, driving real advancements and promising an even faster, more efficient pipeline ahead.
So here's a final thought to chew on. As these digital tools get even smarter, think a more powerful AI, more accurate predictive models, seamless data integration, could we see a fundamental shift in how research is done? Could we eventually reach a point where we can almost, predict and design new therapies with incredible speed and accuracy, maybe even anticipate future health threats and develop solutions proactively? It's a fascinating, maybe even transformative prospect.
It certainly is. And for anyone wanting to dig deeper into any of this, definitely explore the scientific literature. There's a wealth of information out there in this fast-moving field.
Thanks for joining us for this deep dive. We look forward to bringing you another fascinating exploration next time.